thanks, Lucia, for the introduction, and thanks for the invitation to give this talk. So what I'm going to talk about today is that I'm not presenting any of this work. It's not my own work, but I'm presenting work which has been done on the facility, with one exception, that I'm going to show all the work that that I show it has been done on the CLF. So just to very quickly introduce the CLF itself, we're the UK's national laser facility. We have a number of different facilities within the CLF, so including very big lasers, which we see here on the left-hand side, but some smaller scale lasers doing applications really in the physics and life sciences. And the examples I show you today are going to be from this one in the end here, this facility Octopus, which is a cluster of microscopes, of laser-based fluorescence microscopes. All these facilities are available for access by the community. That's why we operate them, and Laserlab-Europe is one of the ways in which you can get access to Octopus and the other facilities within the CLF. So why am I talking about infectious disease? Well, infectious disease has been a problem that's been with humanity since for as long as it's existed, as long as humans have existed. And I think maybe it's been a little bit of possibly a poor relation to some other high-profile diseases, if you like. So we see a lot about, or we have seen a lot about, for instance, about cancer, about degenerative diseases. But over the last few years, obviously, we've had a very unpleasant reminder of that infectious disease is still very much with us in the shape of this thing we see here, the SARS-CoV-2 virus. But of course, it never went away. So there are things like tuberculosis, which actually kills over 1 million people worldwide every year. There are diseases of plants, including crop plants. So there's obviously important for humanity, and of livestock as well. So I'm going to touch on these on all these different aspects during the talk. So if we go, if I look at the I show this from the World Health Organization. This is from a few years ago, actually, before the recent pandemic, and this was diseases identified by the WHO as either presenting existing threats or emerging, or likely to become more important. So you can see along the top here some existing disease threats at the time, and then you see in the second category diseases which were identified as emerging threats, including SARS coronavirus diseases, which obviously was the one that got us. But there's also even unknown things out there that may be out there ready to get us, which we don't even know about, this so-called pathogen X, which the next important infectious agent, which may come along and and cause us problems. So so there's a lot of diseases out there, and the risks are in some ways growing. So if we think about pandemics, obviously we're still in the middle of a pandemic. Hopefully we're getting towards the end of this particular pandemic, but it's still there, and there will be more. It won't be the last, because the risks are increasing. Really, growing populations, there's more and more urbanization, so people are coming closer and closer together, higher population densities, which makes it easier for diseases to spread. Humans are encroaching into new environments. They're coming into contact with animals and other carriers of diseases. Climate change is causing movement of disease vectors and of humans, which again make them come into contact with each other and increase risks. And people travel more than they used to, so it's easier for these things to spread around the globe. So there will be more pandemics. And the other problem, really another big problem in the infectious disease area, is antimicrobial resistance. So the increasing resistance of bacteria to antibiotics. So you can see some quite frightening numbers here. So if we don't deal with the problem, then we could be looking at 10 million deaths annually by 2050, and massive cost to the economy as well as to human life. And in Europe, currently, a significant number of people die every year as a result of hospital infections caused by antibiotic-resistant bacteria. And this is one of the top 10 global public health threats as defined by the WHO It's driven by overuse of and misuse of antibiotics, and by poor sanitation conditions, which allow infectious agents to spread. And it is a major threat to modern medicine. So we rely on the availability of antibiotics to stop infections that can develop as a result of surgery or as a result of cancer treatments. If we can't treat these infections, then those sorts of treatments are under threat. And the other thing I mentioned was climate change. So climate change is threatening global food supply. Obviously, it's a direct threat in terms of more droughts, more extreme weather conditions, but also it's increasing the risk of infectious diseases. So the changes in the climate are increasing the range of pathogens. So pathogens found previously in mainly in tropical areas can spread into temperate regions of the Earth. And the conditions caused by climate change, extreme heat, drought, floods, they can all increase the susceptibility of plants and animals to infectious disease. So in that way, it's also a threat to humanity. So to get onto microscopy, how can microscopy actually help us? Well, microscopy and optical microscopy, laser-based microscopy, is an extensively used technique in the life sciences. And in the area of infectious disease, we can use it to study the biology of pathogens. We can look at how they interact with their hosts, what they do inside the cells, how they get in, what they do once they're inside. We can look at the effect of potential new drug candidates and see how they interact with their targets and how they interact with the organisms which we're putting them into. They can help us understand how our immune system, the immune systems of humans, animals, and plants work to combat infectious disease. And we can actually look at the mechanics of disease transition using these transmission using these techniques. So microscopes can help in a number of ways, and I'm going to show for the rest of the talk a few examples of how they've actually been used to investigate different aspects of disease on the Octopus facility. So firstly, apologies to people who are already experts in the area, but I think as we have I guess a sort of mixed audience, I want to introduce a few concepts just around fluorescence. So these techniques are all fluorescence microscopy techniques that we have in Octopus. So fluorescence is a process which happens when a molecule absorbs energy in the form of a photon. So you can see in this energy levels diagram here, photon being absorbed sends that molecule into an excited state, higher energy state. There's some rearrangement going on here over quite a fast time scale, and then the molecule can actually lose that excess energy, return to its ground state through the emission of a photon, so a fluorescence photon. And because energy is lost to some extent in this part of the diagram, the fluorescence photon is of a lower energy than the photon which went in. So it's a longer wavelength. So we can make use of that characteristic as in microscopy. Something else which I'm going to mention a bit later on is this phenomenon known as FRET, forced resonance energy transfer, and this happens when a so-called donor molecule is close to another molecule, close within about less than 10 nanometers, and in that case, this molecule, when it's in the excited state, can actually transfer its energy to its partner molecule, to the acceptor. And then the fluorescence photon is emitted by the acceptor molecule rather than by the original molecule, the donor. And we can use that to look at interactions between molecules, because it works only over this quite short distance scale, and it's very sensitive to distance. So there's actually a 6th power sensitivity to distance, so it's extremely sensitive to distance, and we can use it to measure distances and look at interactions. And I'll show you an example of that during the talk. So a simple fluorescence microscope would look something like this. So we have a light source, we have something to select the wavelength, and then we have this here, a dichroic mirror, which and all that means it will reflect one particular wavelength but transmit other wavelengths. So this one, so we pick the dichroic mirror to actually reflect the excitation wavelength, the photons which we're putting into the sample. So that light is reflected up to the sample, focused through an objective lens, and then the same objective lens collects the fluorescence, the longer wavelength fluorescence coming back from the sample, which goes back down. In that case, the dichroic mirror transmits those photons, so that goes through some sort of filter to clean up any scattered light, and through onto a detector, usually a camera you can you don't usually look down microscopes through your eyes very much anymore. It's all detected on cameras. So that's the it's the sort of bare essentials of a fluorescence microscope. But they tend to be more sophisticated than that. And I'm going to talk about various flavors of fluorescence microscopy that have been used to look at the different infectious disease problems here on octopus. So firstly, before we start, we do actually need to make our sample fluorescence in some way. So the advantage of fluorescence is that we can specifically label the target molecules that we're interested in. This is why it's become such an important technique for life sciences research. So we can do that in a number of ways. So one of which we can use fluorescent proteins. So these are, you may have heard of GFP. GFP is the original fluorescent protein. This was the one which was found in nature, actually in jellyfish. And these are proteins which are naturally fluorescent, and GFP, green fluorescent protein, obviously from the name fluoresces in the green. But these have been modified over the years to create a whole portfolio of these fluorescent proteins right across the visible part of the spectrum, fluorescing in all sorts of different colors. So what we do if we're using fluorescent proteins is we genetically modify our sample to express a fluorescent protein attached to the protein of interest in the sample. So we end up with a fluorescent marker attached to our protein of interest. So then we can visualize that in a microscope. Other ways we can get fluorescence into a sample is actually by chemical labeling. So in particular, we can label cysteine groups and amine groups within proteins. We can get, and there's a whole range of small fluorescent molecules which are available with reactive groups attached, which will target cysteines and amines. And generally what's done in that case is we label something which will target the protein of interest within the cell. So it could be most commonly an antibody or antibody-like molecule. So we tag on to that molecule a small molecule fluorescent, which is fluorescent, and then we can put those molecules into the cells into our preparation, and we that way label the proteins of interest. We can also attach specific peptides to our proteins of interest, which are then targets of specific fluorescent labels. Again, another way that we can get the fluorescence into the sample. And instead of using small molecules or proteins that are fluorescent, we can also use things called quantum dots, which are a very bright fluorescent nanostructures, which are used in some circumstances, can be the fluorescent marker of choice for our fluorescence imaging. So I mentioned these different flavors of fluorescence microscopy. So I'm going to start with one of the older ones, but something which is still in use every day in very many laboratories. This is confocal fluorescence microscopy, and this was really made possible by the invention of the laser. So you'll see if you look at the picture here, it's quite similar to the very basic fluorescence microscope that I showed a few slides back. But we do have this aperture, pinhole aperture here in front of the light source, and a second pinhole aperture here in front of the detector. And what these two pinholes do is they actually allow us to exclude out of focus light rays. So you can see here, out of focus light rays don't actually get through the pinhole. Only light rays that are in focus get through that pinhole and onto the detector. So that way we can get a lot cleaner images from our samples. And it also enables us to do something called optical sectioning. So we can focus at different depths within a specimen, within a cell, and we can get 3D images by focusing on these different depths. And it's a scanning technique. So we're scanning a spot of light across a sample, detecting the fluorescence as that light beam rusters across the sample. It's picked up on our detector here, and we build up an image that way. And I mentioned this phenomenon of fret. We could actually measure fret in the fluorescence microscope, in the confocal fluorescence microscope, by using something called fluorescence lifetime imaging, where actually instead of imaging simply the intensity of the fluorescence at a particular point, we're imaging the fluorescence lifetime, so how long the fluorescence lasts after it's excited by a pulsed laser source. And that fluorescence lifetime changes when fret occurs. So it's a measure of when fret is occurring in the sample. So we can build up effectively a map of molecular interactions within the cell using this technique, and I'm going to show you something on that in a minute. So so this is really just to illustrate the advantages of confocal microscopy. So you can see here across the top are conventional wide field fluorescence microscopy images, so using the sort of microscope that I showed earlier on. Below we have images from a confocal microscope, so you can see that by cleaning up these out of focus light rays, we can see a lot more detail within our samples, and we can see effectively inside the samples in the case of this pollen grain here by using this optical sectioning technique. So the resolution of the confocal is something like around about 250 nanometers. This is the sort of resolution we can get with this sort of microscope. So I'm going to show you a couple of examples of research into infectious diseases done using confocal microscopy on octopus. And the first one is avian flu, bird flu. This is caused by flu viruses that are adapted to birds. They're a problem in themselves in that they infect and kill birds that are used for food production. They can also spread when humans are in close contact with birds. They can spread into the human population, and human-to-human transmission can then occur. And we're actually going through quite a serious outbreak of avian flu at the moment. This is a strain called H5N8, and it's affecting farm birds and wild bird populations. I just took this from the BBC website from yesterday, talking about anti-bird flu measures which are being put in place here in the UK. But this is it's not just a UK problem; it's a worldwide phenomenon at the moment. This particular group that we had here on the facility were interested in looking at how we might mitigate and stop infections of avian flu. And obviously one way to stop infection is vaccination, and we can give vaccines to birds using modified fowlpox viruses. So fowlpox is a virus that infects birds. It's quite a common disease of poultry. These can be modified so they don't cause illness in birds, but they can be used to deliver vaccines against bird flu. They have been used to some extent, and they have been shown to be effective against preventing illness, but they don't always stop the birds from becoming infected and infecting each other. So so this particular bit of research was actually using confocal microscopy to look at the interactions of the fowlpox virus with the cell and see how it actually gets in the cell and what it does in there. So there are a lot of pictures here, and I'm not going to go through in detail what every one of them is. I don't have time to do that. But what this group were using, they were labeling the fowlpox virus and particular proteins within the fowlpox virus with fluorescent markers with GFP in this case, and they made a fret pair with a protein in kinesin, which is inside the cell. So the theory was that the fowlpox virus actually interacts with kinesin in the cell, and that's a way that actually the infection takes place and spreads. So what we're looking at here, what really the thing to look at is I guess these images on the right hand side and these plots here. So these are looking at fluorescence lifetimes. So when the lifetime is in these orange and green areas, those are short fluorescence lifetimes. They show the existence of fret, so they show interaction of the virus with the kinesin molecule inside the cell. So you can see. So really, what this is telling us is that different proteins in the virus, some of them do interact and show quite a strong reduction in fluorescence lifetime. So they're interacting inside the cell, and that's thought we could use that as a target in a way of modifying these viruses in a way to improve the way that it actually infects the cells and delivers the vaccine. So the other example I'm going to show from confocal microscopy is a plant infectious disease. This is something called potato blight. This is again a disease which has had a major impact in history, and it still emerges periodically. It causes severe damage to potato crops. It was responsible, along with a lot of political decisions that were made at the time, but the sort of primary cause was still this pathogen, which caused major famines in Ireland, and this actually led to very many deaths and wholesale emigration from Ireland at the time because of the large amount of starvation which is going there because of this disease. And it does still it still occurs, and it's still a problem for potatoes and actually can infect other plants as well. So it's a fungal type infection, and the way it invades the cells is in the form of what's called a haustorium. So this is actually penetrating the plant cell wall and is forming this structure within the cell where it reproduces, and this is what results in the damage to the crops and the death of the plant. So the structure, as it's formed inside the plant cell, becomes surrounded by this membrane, the so-called EHM, the extrahaustorial membrane, which is a plant membrane, which is responsible for part of the defense response of the plant. So again, this particular bit of work was aimed at trying to understand a bit more about the defense mechanism of the plant in response to this pathogen. So something thought to be involved is the chloroplast. This is an organelle within plant cells, which its primary role is in photosynthesis, but it can actually also produce antimicrobial compounds in response to infections such as the potato blight infection. So what this group wanted to know was actually what happens to the chloroplast, what do they do in response to an infection. So what they did was they did, again, confocal microscopy here, and tagging the chloroplast with GFP. So you can see the stroma, this is part of the chloroplast, which has been tagged with GFP, and a protein which is associated with this haustorium that's from the pathogen. So they were labeled with two different colors of fluorescent markers, and if we actually overlay these two, what they could actually see was that there seemed to be an association between these haustoria and the chloroplasts. So it's suggesting that as the infection takes place, the chloroplasts are actually brought into contact with the haustoria, and they're coordinating the immune response there. They were also interested in looking at a bit more about how that actually happens, and to do that, instead of labeling the chloroplasts themselves, they tagged actin in the plant cells with green fluorescent protein. And again, this is actually showing interaction between actin and the chloroplast here, and that's actually telling us, so it's believed, that the actin is actually responsible for actually bringing the chloroplast into contact with the haustoria. And that's, so if you disrupt actin assembly with this compound latrunculin A, you actually dissociate the chloroplast from the haustoria. So that's obviously an important factor in the immune response of the plant. And the final bit from that particular publication I'm going to show is using another technique, which you may have heard of, which are optical tweezers or so-called optical trapping. And what that's about is if we take a small object, it is actually possible to trap and hold it in a focused laser beam. So an object in a focused laser beam is subject really to two forces. So scattering force, which will tend to move it in the direction of propagation of the laser beam, but also this gradient force, which is tending to concentrate it into the focused part of the beam. So, and if the gradient force is actually stronger than the scattering force, so if the focus is essentially if we focus the beam strongly enough, we can actually trap and hold these objects inside in the laser beam, and we can do that in combination with a microscope. So we can actually trap objects within a cell and manipulate them and look at what's happening. So in this case, what they did was trap a chloroplast, an individual chloroplast. So you'll see, so if I'll just stop that video, you'll see here, this is an individual chloroplast, and this here is this EHM, the extrahaustorial membrane. So they're actually trying to work out the forces involved in attaching the chloroplast to the membrane. And if I let this run, what they actually did was try and drag the chloroplast away from the membrane and see what happens. So you'll see it move away, and then you'll see it spring back. And you'll see it again here. So what that's actually telling you is that this the chloroplasts are in fact attached quite strongly to the membrane, strongly enough that the force of interaction can actually break the optical trap, and we can actually get quantitative measurements there of how strong interactions are between components within cells using this optical trapping technique. So now I'm going to move on to a few other microscopy techniques, in particular techniques that have become known as super resolution optical microscopy. So optical microscopy is diffraction limited, and this was first stated by Abbe in the 19th century, and you might be familiar with this equation, which is telling about really what we can resolve in a microscope. So this number D here. So and the important factor here is the wavelength. So we're limited by the wavelength, and approximately half the wavelength of the light. So conventional optical microscopy, we can get into the as I showed you with the confocal technique. The best we can do is in the sort of few hundred nanometers, about 250, 200 nanometer resolution. But in recent years, a number of techniques have been developed to actually get around this, these so-called sub-diffraction limit imaging techniques, or super resolution. And to 2014, Nobel Prize was awarded to these three people who developed different super resolution microscopy techniques, and these are letting us actually get down closer to the molecular level inside cells. So what I'm going to show again are a few examples of research on the Octopus facility using some of these super resolution techniques to look at infectious disease. So the first one is I'd say sort of half super resolution technique. It's half and half defined as being really a super resolution technique. I'm going to go to the true super resolution techniques next, but this one is called structured illumination microscopy, SIM. And the way this works is we illuminate the sample with a pattern of light. So and then what we're doing is looking at interference between the sample. So the sample has spacings within it which we don't know. The illumination has spacings in it which we do know. We're illuminating with a particular pattern from a diffraction grating. And we can actually get interference patterns from these two when they're together. So so you'll all be familiar with these moire fringes, interference fringes that you see. And in microscopy, we can actually use quite a bit of computation to actually reconstruct from these fringes, which contain information at sub-diffraction limit resolution, and reconstruct an image from that. So so just a quick example here. This is just of a group who are in the same building as us, and this is really a test image just showing chromosomes. You can see here conventional wide field image here, a SIM image on the right. So I think you can see there the improvement in resolution from the SIM technique, and it's got a resolution of about 100 nanometers. And on octopus, this has been used to look at an infection called toxoplasmosis. So this is a disease caused by a parasite. It's so-called EP complexum parasite, and it infects a lot of people worldwide. So it varies a bit from region to region, but up to 50% of the global population believed to be infected with this parasite. Now, generally, people can be infected with this and not even know about it. It doesn't cause any problems, but it does cause problems in pregnancy. It can cause, in particular, eye problems for the developing fetus, and for people who have suppressed immune systems, in particular with HIV, this has become a problem. So you can see here, this is one of the this parasite goes through a number of different stages in its life cycle. This is taken from the it can infect nervous tissues. Is a target tissue for this parasite. And you can see here from cerebrospinal fluid, we can see some of these other parasites here. So the life cycle, the definitive host of this parasite is the cat. So the cat infected with toxoplasmosis sheds these so-called oocysts in the feces, and these can infect. Either they can infect the so-called intermediate host, which is mice, birds, but also they can infect humans directly if they come into contact with cat feces, and as with poor hygiene, they can infect people. But they can also infect animals which humans eat, and then if humans eat poorly cooked meat from an animal which has been infected, you can get infected that way. And the cycle repeats itself because the infected mouse can then be eaten by a cat, which then picks up the infection. So this is how this disease transmits. An interesting evolutionary curiosity, there was a paper came out from a year or two back showing that this the parasite, the infection, actually changes the behavior of mice. So mice infected with Toxoplasma actually lose to some extent their fear of cats. So they are more likely in that way to be eaten by the cat, and that way the parasite transmits itself. So it's in that way it's evolved to transmit more effectively. So a group here have used SIM to look at Toxoplasmosis and see how it actually replicates. So one of the questions that people were interested in finding out is, when this parasite actually replicates within the organism, what happens to these organisms called micronemes, which are organisms which actually facilitate the parasite getting into the cells. So a parasite will multiply within the body, form these, so a single mother cell will form these so-called daughter cells, which then go out into the organism. They will then reproduce and form another mother cell, and that way the parasite reproduces and develops within the infected host. So what they wanted to know is what happens to these micronemes. Are they actually retained within the original mother cell, or do they actually get distributed to the daughter cells as the parasite reproduces? And the way they looked at that was actually taking labeling a microneme protein MIC2 and labeling it with two different colors at two different time points during the infection process. So at the beginning with this di-TMR, and then later on with the second ISIR. So you can see here a single cell, which is then going to two cells. You can actually see one cell on the top here and on the bottom, and then going into this 8-cell structure here. And so the conclusion from this work, if you look at the actual these sort of magenta dots here, and this is the originally labeled protein, you can see that it does actually get distributed to the different cells as the virus, the parasite reproduces. And you can actually count that. This is actually showing the number of micronemes in the individual daughter cells. So that was one conclusion from this work. Another thing they were interested in is actually what happens to these micronemes. How do they get to the right place on the parasite to be able to infect the host? And in this case, again, more SIM imaging here. You can see these quite nice high resolution images, and they also labeled actin again within the system. You can see here these orange colors are actin, and orange was shown to be associated with the microneme. So the actin is actually associating with these recycled microneme vesicles, and that way it is transmitted around the group of cells and enables the parasite to infect effectively. So the other super resolution technique I'm going to mention is so-called single molecule localization microscopy. This exists again in a couple of variants, so-called STORM and PALM, but they're essentially the same. They're slightly different in detail, but they do the same sort of thing. So this microscopy technique relies on being able to image down to the single molecule level. So if you image a single fluorescence emitter in a microscope, it will appear as a blob because of diffraction. So it will show as what's called the point spread function of the microscope. So as the name suggests, it's how much a single point is spread out when you image it within a microscope. But what you can actually do is, if you can collect enough photons, if you get a good enough signal to noise, you can actually locate where that individual emitter is within that blob of light. Essentially, it's located at the center of that blob. So by fitting to that, you can actually localize the molecule to much better than optical resolution. So just to show how that works, this is a video taken on the Eiffel Tower with lights, which had at this time certainly different lights which flashed on and off at different times. And that's how we would do these imaging techniques. So instead of lights, we'd have individual fluorescent molecules which are switching themselves on and off at particular times. And as time goes on, we detect each one of these individual emitters and then actually determine where those are actually located. So if I run this, you'll see detection of particles going on here as the lights are flashing on and off. And you'll see at the end the reconstruction, which is the high resolution image, which is built up from the detection of lots of these individual single molecules. So again, just to show you here, this is just an example which I took from a paper. You can see a conventional fluorescence image and a STORM image here. And again, you can see how much better the resolution is with this technique. And with this method, you can get to around about 20 nanometer resolution. And this was used to look at this infection. This is a bacterium Chlamydia. So Chlamydia it's most commonly known as a sexually transmitted disease. But there's another bacterium, Chlamydia Pneumonia, which is actually causes respiratory infections. And it's quite a common infection found. If you test people, we can actually find antibodies to this particular bacterium in quite a number of people. So acute disease is one problem which you can treat with antibiotics, but it's also implicated in chronic and inflammatory processes. So infection in the lungs can spread throughout the body and can ultimately result in arteriosclerosis, so results in heart disease. So it's quite important to understand a bit more about this particular pathogen. And the group here, we're trying to look at this outside of this, the pathogen is covered with a membrane, the outer membrane complex of proteins, which is quite important for the survival of the bacterium and for the infection process. But it's not really known how that goes together. So they were looking at a protein commonly found, which is this so-called MOMP protein. And the theory here being that this protein has a lot of cysteine residues in it. So the theory was that it actually uses disulfide bridges to build this protein complex. So this work here using STORM microscopy, they mutated a number of cysteines to alanines, actually, to break that any disulfide bonds that might form, and looked at the effect of these different mutations on the clustering of this molecule within the membrane of the bacterium. So you can see here, it may not be terribly obvious from some of these images, but you can sort of see in the wild type, you can see these sort of, you can see quite a few sort of dots, speckly sort of appearance, and in some of the mutants, it's maybe a bit more widely spread out around the bacteria. And if you actually take those images and analyze them, do a cluster analysis, you can see that in some cases, some of these mutations are actually reducing the clustering of this protein within this complex of proteins. So it's telling us something about the importance of these different cysteine regions. And it appears to be the case that maybe more than one of these is involved in forming bonds and holding this structure together. So the final super resolution technique I'm going to talk about as I'm sort of coming to the last 5 minutes or so of the talk is this technique STED. And this is if you look at this picture, it's a little bit like the confocal technique in a way, in that you're scanning a spot of light across the sample and building up an image that way. But in the confocal, you're back to the diffraction limit again. The size of the spot that you can scan across the sample is limited by the wavelength of the light. But using this technique, we can actually use a second laser, and we can engineer the beam of that laser to have this sort of donut pattern, which overlies the scanning spot, the fluorescence excitation laser. And if we pick our wavelengths right, and we pick our fluorescent markers right, this second so-called STED beam will actually switch off the fluorescence in the area around that's covered and just leave the fluorescence coming from that hole in the center. So what we're effectively doing, this is making the spot that scans across the sample smaller than it would be in a confocal microscope. And again, just an illustration here of the improved resolution, conventional and the STED image here. And this can get us about 50 nanometer resolution. This was used to investigate some potential new antimicrobial drugs. So I've just shown here a few. I'm not going to read through all these, but just to show a few drug-resistant bacteria which are becoming a problem for humans. So things like E. coli and Staph aureus are increasingly resistant to antibiotics. So we need to find new ones. And in particular, these so-called gram-negative bacteria, there are not as many antibiotics that target these. So we need to find effective new antibiotics against these, which work with different mechanisms from conventional antibiotics and can somehow overcome resistance. This group looked at these Ruthenium complexes, they're potential antimicrobials. They were developed really as cancer therapies, and it so happens that they are luminescent, so they will actually fluoresce themselves within cells, so we can image them quite easily without using additional imaging probes. And they were known for some time to have some antibacterial activity, and they're known to be effective against both gram-negative and gram-positive bacteria. And you can see here what these graphs are showing is how effective they are at killing. So it's killing these different types of bacteria. So as the number goes down with time, this is showing the bacteria actually being killed by this particular complex. So what they wanted to do was understand a bit more about the mechanism of action. So to start off, they use SIM imaging, and they're actually looking at, with time, where these complexes go as they go into the bacteria. In E. coli was the system that they used. And we get these from these sort of more spread out appearance at the start here to these more sort of concentrated images as time goes on. So this was thought to be typical of them accumulating within the nucleus. So we went to the higher resolution STED technique to actually try and look at what was going on within the nucleus and see whether that really was the case. So what we're looking at here are images of, in the green, this is DNA labeled with a fluorescent marker, and the red is the ruthenium complex. So you can see in these different images here, they're overlaid. When you overlay these images, the DNA, the ruthenium complexes are clearly attaching themselves to the bacterial DNA, and this is how they're actually killing the bacteria. So finally, I'm gonna really step up a length scale and talk about something which has become, obviously we see a lot more of these around these days, and we've seen a lot of them over recent couple of years. So we can use fluorescence techniques to help understand really how we can stop infection actually happening at the even before it actually gets to the cells. So this is a scanning electron microscope image of what one of these masks actually look like. So it's made up of these layers of fibers. So we're actually using laser tweezers to trap droplets. So airborne viruses like SARS-CoV-2 spread on droplets. So we're actually looking at using these laser tweezers to investigate how droplets interact with these masks, how we can actually improve masks to stop them getting make it more difficult for the droplets to get through. And we can use fluorescence microscopy techniques to look at how viruses are actually distributed within these sorts of droplets. This is work which has only just really got underway here. It's part of this study, the PROTECT study, which is looking at all sorts of different aspects of COVID-19. And this is being led here by my colleague Andy Ward, who kindly supplied these images before publication. So it's work in progress, but hopefully we're going to understand a lot more about how viruses actually go into our bodies as passengers on these droplets. So the last two slides, I'm just talking maybe a little bit about where I see the future in these sorts of techniques for studying infectious disease. This example here I'm going to show here, this is the one example that I have which wasn't actually taken on this facility, but there's still relatively few and far between using correlative microscopy. So this is something called CLEM, correlative light and electron microscopy. And the advantage of those two techniques are fluorescence microscopy obviously gives us the specificity. We can use fluorescent markers to label the molecules of interest, but electron microscopy gives us really very high resolution structures. So we can actually prepare samples in a way we can image them with both techniques. So we can grow cells. We can grow them on these grids, which have grid squares, which are each individually annotated. Then we can image these in a fluorescent microscope. We can see green here, which is where fluorescence is actually occurring. We can then embed these in a resin suitable for electron microscopy and image them with an electron microscope. And we can use the grid reference markers to actually co-locate these regions. So you can see here, this is just an example of, this is really a test system, something called Semliki forest virus. And look at infection of cells. And you can see here, this is an overlay of fluorescence microscopy with electron microscopy, and it's telling us, showing us where the viruses are going inside the cells, and we can actually get down and look at what's going on really at the molecular level inside the cell using electron microscopy. So so again, I think we'll be seeing a lot more of this this technique in the future. And the final thing I'm going to show, and this was actually from octopus, but doesn't actually have any optical microscopy in it. This is so we do, as well as laser microscopy, also have some electron microscopy here, in particular something called FIB-SEM, focused ion beam scanning electron microscopy. And this can give us 3D images of samples with quite high resolution, and we can do it under cryogenic conditions. So we, instead of using resins to fix our samples or chemical fixation, we can actually freeze them. And we used this was a collaboration with some other people here on the campus, combining this technique with transmission electron microscopy to actually get a lot more information about how the SARS-CoV-2 virus infects cells, what it does to cells when it goes in there, where it goes, how it interacts. And there's quite a lot of information came out of that paper. I don't have time really to talk about it now, but it's the reference here if anyone's interested. But they do really show the damage that this virus can do to the cells. So my time's about up, I think. So so I'm just going to conclude. Hopefully I've shown you that laser fluorescence microscopy can give us insights into really a whole range of aspects of infection and immunity. I've only used examples really from our facility, but this is going on across the world, in many labs using these techniques to look at infection. If we go to these super resolution techniques, we can actually start getting down to the molecular level and see what's happening between pathogens and cells on that level. And I think we'll see more use of these correlative methods. So let's combine this molecular specificity of fluorescence with ultrastructure from the electron microscopy. And really just a note for the future, and something that we're trying to do here, if we fix things cryogenically rather than chemically, that actually makes our correlative microscopy job a lot easier. But it's still difficult to do super resolution techniques at cryogenic temperatures, and that's a priority really, to get closer in resolution between the optical technique and the EM technique. And that's something we're working on here at the moment, and hopefully we'll stop. That will become available. We're currently offering cryogenic super resolution technique, the STORM technique, to users. We hope to use that in a correlative mode quite soon. So thanks for listening.